So today's topic is mutation. We are starting molecular biology. Hopefully, if we can finish tomorrow and day after tomorrow with the classes, those portions will be included in the examination. If we cannot finish, they won't be. Right? So mutation, already you know what is mutation. Mutations are something which is a change or alteration in nucleotide sequence in nucleic acids, which is transmitted to the next generation. If the change has taken place but not transmitted to the next generation, it is not called mutation. The best example of this is the change or alteration in RNA. Since our genetic material is DNA, unlike RNA virus, any alteration in RNA is not transmitted to the next generation. Somatic mutation is something which is locally oriented. Which occurs in only one generation. So, for example, skin cancer due to exposure to high UV radiation. Okay, this skin cancer will not be propagated to the next generation, but some other cancers like cervical cancer may propagate from one generation to another because it may be transmitted through germ cells. What are the causes for mutation? Inherited and acquired. Acquired mutation may be due to environmental damage, say consumption of carcinogenic compounds, application of carcinogenic ointments, accidental, or spillage of radiation. Spillage of radiation. Say for example, Hiroshima, Nagasaki, Fukushima, right? And Chernobyl in Russia. Few good examples. Mistakes when DNA is copied. This occurs, is a normal process. When DNA is being copied, the mistake will take place. In some people's body, it will be rectified properly. In some people's body, it won't be rectified properly. Those who will fail to rectify it proper way, they will carry it as a mutation. But you can say that if the DNA is mistakenly copied, how come that can be further copied to next generation? So this is a type of repair called SOS response, where this is the motto that it is better to survive with some loss than getting extinct. কি বলছে SOS response? SOS response বলছে when DNA is being copied and when they come across a small mistake and there is no time that it can rectify the mistake. Because now if they do not continue the copy, making the copy of the DNA, the cell will die. So what they do, they randomly put some nucleotide there with a chance of 25% being mistaken. They continue the replication process. In this process, it is called erroneous process. Although it's a type of repair mechanism, but 75% chance that repair will be with mistake. Out of 4 bases, ATGC. So one mistake, 25% chance it will be correct. 75% chance it will be wrong. But still, this is followed when, say, when we are irradiated with huge amount of radiation all over skin. What happens? Then if all the skin cells stop doing the replication, what will happen? Immediately we will die. But that never happens. We suffer from burns, we suffer from exfoliation, some skin problem, and then we recover. That means some or other cell continue the replication process. But while doing so, they do it with lots of mistake. With this in their mind, the cell, that it is better to survive with some loss rather than getting completely extinct. Types of mutation. There are two types of mutation called gene mutation and chromosomal mutation. Gene mutations can further be classified into point and frame shift mutation. Point mutation can be of two types: substitution. Substitution is one of the type point mutation. Substitution means, suppose instead of adenine, thymine or guanine will be placed. Right. Now, we're taking this as a template. When we are synthesizing this strand, instead of A, instead of T, they might put A, they might put G, they might put C. There's a mistake, right? In the next cycle of replication, in this cycle is okay, fine. 50% will be wrong, 50% will be right. In the next cycle, this will be considered as a template, and then the mistake will be incorporated. So when instead of A, if substitution is with the same group called transition, that means purine to purine or pyrimidine to pyrimidine. Means here it was supposed to be thymine, correct? If it is changed by adenine, then it is pyrimidine to, sorry, if it is changed with cytosine, then this is pyrimidine, this is also pyrimidine, so it is called transition. If it is changed with, say, guanine or adenine, then it becomes transversion. Clear? Clear? Purine to purine, pyrimidine to pyrimidine, transition. And crisscross purine to pyrimidine or pyrimidine to purine is called transversion. So usually this is shown by this, like this. Okay? So horizontal lines are transition. And cross lines are transversion. Like this you can also show this. Now, what was the beginning to this transition and transversion? Substitution. That means what? Suppose this had 10 base pairs. The faulty template also will have 10 base pair. This is called substitution. Means one has been replaced by another one. Fine? In that case, there are 2 points: transition and transversion. But if that is completely deleted, then or a new nucleotide is added, then it is called deletion or addition. It might happen so that while replication, this was removed completely and the sequence became like this, correct? This might happen, then it will be deletion. And if a new, which was not there, is added, then it will be called addition or insertion, you can say. Now, if due to this deletion or insertion, if the reading frame for translation process is altered, then it is called frame shift. Reading frame may not alter because all the portions of DNA are not read. Only 1% of the total DNA is actually responsible for all the proteins we express. Less than 1%. So it is a less than 1% is responsible for production of all proteins of Homo sapiens, and rest 99% is responsible for making us Homo sapiens. Only 1% is responsible for proteins produced by Homo sapiens, and more than 99% is responsible for making us different from cats, dogs, fish, oyster, whatever it is, right? 99% is not coded, not synthesize any protein from that part, but that part is not inert, that has got evolution-wise importance. That tells the story how we got changed from different apes, and not by any days have changed, we can't take their names. Harry Potter, the one we do not speak about, so we cannot speak about, the days has come. So we are not produced, we are synthesized. Not by him, someone else, whom we do not worship. Definitely, we do not worship. Fine. So, that 1% is responsible for synthesis of all proteins. Say, for example, hemoglobin A1, hemoglobin B, hexokinase, pepsin, chymotrypsin, endorphin, everything. That 1%, if anything goes wrong, then we get some disease, genetic disorder, like collagen disorder, like lactose intolerance, etc., etc. Similarly, that part may not produce any protein, that even though that 1%. If there anything happens, it will not change anything. But in a reading frame, if it happens so that the whole reading frame is changed, then it is called frame shift. See how insertion and deletion can change the meaning of the sentence. This is an example. The fat cat ate the rat. Now, what happened? You inserted one H. The meaning changed. Meaning changed. That means genetic message so that the protein may not be able to perform its function. So, protein hemoglobin is changed to some other form which cannot transport oxygen so efficiently. Deletion. This H was deleted. What happened? So, does this have any meaning? What do you call this protein then? A protein which is formed like this is called garbled protein. Garbled protein. That means it may be synthesized like a polypeptide, but without proper biological function. Now, here what is happening? Substitution. Deletion and addition I showed. Now, so substitution can have three types of effects. In place of another. Substitution can have three types of effects. One is silent, nonsense, and missense. What is the meaning? Silent means the change has occurred in the codon which codes for same amino acids. What do you call this process, property of codon? Degeneracy. There are many amino acids which are coded by multiple codons. So, a change in the codon may have taken place in such a way that the ultimate amino acid being produced remains same. In that case, it is called silent. Silent. So, it was like, you see, TTC, it became AAG and it became normal one. Mutation AAG and AAA both codes for lysine, so it became lysine. Right? AAG and AAA both codes for lysine, so it became lysine. So there will be no change in the shape, structure and function of the polypeptide. So it will be silent. If the change takes place in such a way that one of the intermediate codon becomes stop codon, then what will happen? A truncated protein will be produced, or polypeptide. Means suppose it was supposed to contain 100 amino acids. Now with this introduction of stop codon in between, now it is producing only 60. Right? Shortened. So this will have no function, biological function. So that will become nonsense with no biological function. And if that happens so, like this, instead of AAG, it becomes AGG. But problem is not that big. How? What was the targeted amino acid to be? Lysine. What we are getting here? Arginine. What is the difference? Both are basic amino acid. So probably some of the function of lysine might be done by arginine. Correct? Then this will be missense. This will be missense, slightly reduced function, but not fatal. Slightly reduced function but not fatal. It can be threonine. Threonine can also function to some extent, but again it may not be fatal. If it is fatal, then it will become nonsense. This part will always be judged by the phenotypic expression, how the effect is coming in the surface. So keep this, remember this, remember this, okay? It's very important. Very often they ask. And this particular chart is given in Harper and also in many of the books you read. Whenever this kind of mutation, types of mutation comes, you must write. When it comes to what? Point mutation, then you must write this, not frame sheet mutation. When it comes to point mutation, you must write this. 3 types of hemoglobin. Hemoglobin Hickory, HBS or sickle hemoglobin, and hemoglobin Boston or HBM. This is unacceptable missense, and this is partially acceptable missense, and this is acceptable missense. Say, go back to the previous slide. See, arginine and threonine. Lysine is replaced by arginine. Same, both are basic amino acid. But with threonine, something is changed. What is that? Arginine is positively charged. Threonine is probably negatively charged, OH. It can act as a proton acceptor, right? So this change will be further less active. So this part is again coming under missense. Nonsense, silent, missense. Missense can further be, okay? So now you make the whole tree of the mutation. Find out. Part of the tree is done. Only this part is done, right? Yes. Tell me, what is the next? Oh, okay. Point and right? Hello? Now, what is next? Which one? Substitution. What is the next classification? Huh? Fine. Then these two. Follow? Based on phenotypic effect, insertion or deletion can lead to or substitution. Which one? Correct? It may or may not cause frame shift, substitution, these two. Acceptable means, no, sorry, that way, na? Silent, nonsense, makes sense. আরেকটা কি আছে? আচ্ছা, fine. This example, remember, hemoglobin boson, partially sickle hemoglobin, and that is hemoglobin hickory. HBM na? HBM na? These is a little bit confusing, so try to remember like this. Okay, for rest half is further more complicated. Fine. Up to this, okay? Can you remember this chart? Huh? This chart you have to remember. This one. Very often they ask. Very often they ask. These three examples you must remember. Chromosomal mutation, I told you it is more complicated. Deletion, inversion, translocation. First you remember the names, types, okay, and non-disjunction and duplication. Deletion, ball, deletion, inversion, translocation. What does it mean? What comes to your mind when you hear about deletion? কোথায় মুছবো? এদিকটা মুছি? What is chromosome? Right? Okay. What do you call these? Chromatids. What do you call this region? There are 4 dots here, dense areas in the centromere. What do you call them? This is kinetochore and this is centromeric chromomere. Centromeric junction points. Okay. Now, deletion means what? When this chromosome is replicated, then like this is formed normally. Suppose this part is lost. Huh? A part of the chromatid is completely lost. So it is deletion, inversion, when it exchanges, translocation from here to here or here to here, non-disjunction. It did not separate properly, remain together. Duplication, increased in number. One portion joined the other way. It happens after the replication is over. Duplication. This had 1 copy. Here there are 2 copies. Example, duplication of gene, alpha thalassemia. The alpha has got 2 sets of copies, 2 pairs of genes. Translocation. It was like this. A part of it here to here, here to here. Side by side. This exchange may take place within non-sister chromatid as well as sister chromatid. Translocation. During cell division, if they do not separate. Have you heard of trisomy, nullisomy? This is the effect of this chromosomal non-disjunction. One of the gamete don't receive a copy at all. Like this, see, red and blue, it should be 1 red, 1 blue. Now it has got 2 blue, 1 red and no blue here. So this becomes N1, this becomes N plus 1. So when it combines with normal N, what will happen? This will become trisomy and this will become nullisomy. Significance of mutation. Most of the mutations are neutral, except few. Here are some diseases mentioned which are caused due to mutation. See, are they harmful? No. Hrithik Roshan, blue eyes, right? Good looking. Not harmful at all. Aishwarya Rai, green eyes or blue eyes, something, blue eyes. Nothing wrong, right? It's a plus point. You know? Whereas a birthmark, you know, some big patch, birthmark, it becomes good or bad. For police, it becomes good. It becomes an identifying mark. You know? When you commit a crime, it will be easy to identify you. You know? And when you do not commit a crime, it will be difficult to find the partner of your crime. Understood? Marriage problem. Who will marry you? If the big patch of black patch is there on your face, and who will marry you? Huh? No. It's simply you studied all those poetries, poems. The beauty lies in the eyes of the beholder. It is the beauty of the mind, not the beauty of the face. Huh? Just for saying also, huh? Oh. See, this is the real world. Fine. Some are harmful. Sickle cell anemia. Now, sickle cell anemia is boon in disguise. For whom? For the people who live in the particular region, where malaria is predominant. Because they survive, those who are having sickle hemoglobin, they survive from malaria attack. So it is a boon in disguise. But this is not. Correct? Then what is the effect? Bad effect they get with sickle hemoglobin? Anemia. Then a persistent anemia. Very often they might be given, if the disease is very severe, they should be given a blood transfusion. Some are beneficial. This effect is beneficial. Another is, if immunity occurs in some ethnic group, it has been found that the occurrence of HIV is very less. They found that they are somehow immune to HIV infection. It happens. You can try whether you are immune or not by injecting virus. Should you try? Try করা উচিত? উচিত না তো? Causes for mutation. চটপট লিখে নাও। Spontaneous chemical changes. Depurination. Right? Depurination. How? There are enzymes called AP endonucleases. They search for those AP sites, A purinic site or A pyrimidinic site. Wherever they get it, they think that there is only loss of only one purine or one pyrimidine. They replace the counterpart with only one, but actually the loss of two purine or two pyrimidine, mostly two pyrimidine. But they think that it is loss of only one purine. So they replace by only one purine. And by doing so, it inserts mutation. Deamination. Certain compounds, chemicals are there which causes deamination. Give me one example where deamination changes the base. বল, এই যে, হ্যাঁ তুই বল। ডিঅ্যামিনেশন হলে বেস চেঞ্জ হয়ে যায়, একটা এক্সাম্পল দাও। এনিওয়ান পারবি? তুই পারবি? কে হাত তুললো? কে হাত তুললো? তুই হাত তুললি? তুই পারবি? বল, হ্যাঁ বল বল। সাইটোসিন টু ইউরাসিল, ভেরি গুড। সাইটোসিনের মিথাইল গ্রুপ যদি সরিয়ে দিই তাহলে কি হবে? ইউরাসিল হয়ে যাবে, হ্যাঁ, ডিঅ্যামিনেশন। ভালো, গুড, ভেরি গুড, সোনাম। ফাইন, এক্সাম্পল অফ ডিঅ্যামিনেশন। কালকে তো ছুটি নাকি? আজকে তো আমার 5টা পর্যন্ত ক্লাস করতেই পারি। আজকে 5টা পর্যন্ত ক্লাস করতে পারি না? কালকে তো আসবি। আলফা, বিটা, গামা, ওরে কালকে আসতে বারণ করেছি, আজকে নয়, এখনো ছাড়িনি। ফিরে আয়, ফিরে আয়। শিয়ালকে ভাঙাবার দেখালেই হয়েছে। ফিরে আয়, দেখ, আলফা, বিটা, গামা, এই 3 ধরনের রেডিয়েশন আছে। এর মধ্যে কোনটা হার্মফুল বলো? গামার মাস কম বলে পেনিট্রেশন বেশি। এক্স রে দুরকম হয়, হট আর কোল্ড। কোনটা ক্ষতিকারক বেশি? হায়ার ওয়েভলেন্থে এক্স রে ক্ষতিকারক বেশি, মোর হার্মফুল। ঠিক আছে, ইউভির মধ্যে 254 to 260 এটা ক্ষতিকারক, হার্মফুল। ঠিক আছে, আর আয়নাইজিং রেডিয়েশন কি আছে থাকতে পারে? যেমন কসমিক রেডিয়েশন, ঠিক আছে, কসমিক রেডিয়েশন থাকতে পারে। আর যদি নিউট্রিনো রেডিয়েশন হয় কি হবে? এই তুই বল, কি এই নাম তোর? না ওই যে পিছনে, দাঁড়া, না তুই দাঁড়া, হ্যাঁ চাল, চাল, দাঁড়া, চাল তো না, আম, কি নাম? ঘটক, অর্ক বল, হ্যাঁ বল। নিউট্রিনো রেডিয়েশন হলে কি হবে? নিউট্রিনো রেডিয়েশন কে বলতে পারবে? বল। কিছু হবে না কেন? জোরে বল। বাহ, সাবাস, সুপার ছে, বলতে পারো। তাহলে ওটা খুব ছোট বলে ফাঁক দিয়ে বেরিয়ে যাবে। বসো। তুই দাঁড়া, ম্যাটার, দাঁড়া। এবার বল, পজিট্রন এমিশন হলে কি হবে? হ্যাঁ কি হবে? ক্ষতি হবে কি হবে না? এতে যাওয়া হবে না, এটা বড় বলে ধাক্কাকে সরে যাবে। বড় বলে ধাক্কাকে সরে যাবে। আলফা হলে কি হবে বল? আমার দ্বারা ম্যাটার। আলফা রেডিয়েশন কি হবে? ঘন্টা হবে, ঢুকতেই পারবে না। বিটা সুপারফিশিয়াল, গামা ইন্টারনাল। এটা দেখো, থাইমিন ডাইমার ফর্মেশন হয়, মানে যে মিউটোজেন কি তৈরি করে? এই মিউটোজেন দিয়ে দুটো পাশাপাশি থাকা দুটো নেইবারিং পিরিমিডিন বেস একটা কাপল তৈরি করে, তার মধ্যে একটা মানে কি বলে 4টে কার্বন দিয়ে একটা স্ট্রাকচার তৈরি করে, তাকে বলা হয় থাইমিন ডাইমার বা থাইমিন সাইটোসিন ডাইমার। হয় দুটো থাইমিন থাকবে, নয় একটা থাইমিন একটা সাইটোসিন থাকবে। মোটমাট পিরিমিডিন ডাইমার তৈরি করবে। পিরিমিডের ক্ষেত্রে হয় না, পিরিমিডিন ডাইমার তৈরি করবে। হয় দুটোই থাইমিন, নয় একটা থাইমিন একটা সাইটোসিন। এই ডাইমার যখন থাকবে তখন ডিএনএ পলিমারেজ এই ডাইমারকে চিনবে একটা বেস হিসেবে, তার এগেনস্টে একটা বেস বেশি চলে যাবে। তাহলে কি হবে? ডিলিশন হয়ে যাবে, তাই না? কেমিক্যাল মিউটেজেন, সার্টেন বেস অ্যানালগ, এই যে যেমন 5-ব্রোমো ইউরাসিল বা 5-ফ্লুরো ইউরাসিল, 5-FU, এরা কি করে? এরা এই বেসগুলোকে জায়গায় রেপ্লিকেশনে পার্টিসিপেট করে, করে রেপ্লিকেশনকে ফার্দার হতে বাধা দেয়। তাহলে কি প্রপাগেশন অফ রেপ্লিকেশনে স্টপ করবে, প্রিভেন্ট করবে। তাহলে কি হবে? রেপ্লিকেশন হবে না। ক্যান্সার সেল এবং এমন কিছু সেল যারা খুব র্যাপিডলি মাল্টিপ্লাই করে তারা মারা যাবে। সুতরাং এই ড্রাগগুলোকে আমরা ব্যবহার করতে পারি কি? বিনাইন হাইপারট্রফি অফ প্রস্টেটের চিকিৎসার জন্য, সোরিয়াসিসের চিকিৎসার জন্য, আর ক্যান্সারের জন্য। মডিফাইং এজেন্ট কি কি থাকে জাস্ট নামগুলো লিখে রাখো। ডিঅ্যামিডিং এজেন্ট, হাইড্রক্সিলেটিং এজেন্ট, অ্যালকাইলেটিং এজেন্ট আর ইন্টারকাইডিং এজেন্টস। এগুলি কোথাও বাদ দাও। আচ্ছা, এগুলি বেশিরভাগ খাবারের সঙ্গে আসে, সবগুলোই খাবারের সঙ্গে আসে। আমরা যে খাবারগুলো খাই, ফুড কালার, ফুড টেস্ট বাড়ানোর জন্য যেগুলি দেওয়া হয়, অ্যাড করা হয় অ্যাডিটিভ, প্রিজারভেটিভ, তার মধ্যে এগুলি থাকে। কেন প্রিজারভেটিভ থাকে? কেন প্রিজারভেটিভ দেওয়া এই জন্য যাতে ব্যাকটেরিয়া বা ভাইরাস না জন্মায়, কিন্তু ওরা ভুলে যায় যে ওটা আমরা খাবো। In order to increase the shelf life of certain food products, they add these beyond the permissible limit. And in our country, the laws are very old. They do not bother because we have plenty of people to die for. If suppose 50 crore people die in India, nothing will happen. India will still be #1 in population. 50 crore means 1/3. 2/3 is survived. So we don't bother about the death of the people in India. Do we? No. যদি করতাম তাহলে আমরা আইসক্রিম খেতাম না, আমরা বিরিয়ানি খেতাম না, আমরা কাবাব খেতাম না, বাচ্চাদের তৈরি হয় আর কি। আমরা ফ্রোজেন চিকেন, মাটন এগুলি খেতাম না, আমরা পোলট্রির ডিম খেতাম না, আমরা আর্টিফিশিয়ালি চাষ করা মাছ খেতাম না, ক্ষেতের থেকে তুলে আনা নন অর্গানিক শাকসবজি খেতাম না, আপেল, আম, লিচু, পেয়ারা, বরবটি, পুঁই শাক, শিম, ফুলকপি, বাঁধাকপি, কুমড়ো শাক, কুমড়ো, লাউ কিচ্ছু খেতাম না। চাষিরা এগুলি নিজেরা চাষ করে, নিজেরা খায় না, আমাদের খাওয়ায়। নিজেরা কি করে? এইগুলি ছাড়া চাষ করে নিজেরা খায়। আর আমরা কি খাই? এইগুলি দেওয়া খাই। নাইট্রাস অ্যাসিড এজেন্ট, অ্যালকাইলিন এজেন্টের নামটাকে মনে রাখো যদি মনে রাখতে পারো। কম্পাউন্ডের নামগুলো মনে রাখো জাস্ট যদি মনে রাখতে পারো। ইন্টারকাইডিং সাবস্টেন্স, ইন্টারকাইডিং সাবস্টেন্স মানে these compounds are used mostly for visualization of nucleic acids. আমরা যখন একটা ডিএনএ কে when we separate the DNA based on their size in a gel slab by gel electrophoresis, then we must find out the proper fraction of the DNA. How to visualize? We must add certain intercalating, certain stain How to stain them? Because proteins take up stain, they become colored. But nucleic acid do not put up any color. They should be given certain compounds which can intercalate, make space between two bases, and which gives color upon irradiation by কি বলে এটাকে luminescence or phosphorescence, তাই না? luminescence or phosphorescence. তাহলে যদি আমি যেমন অ্যাক্রিডিন অরেঞ্জ, ইথিডিয়াম ব্রোমাইড, ঠিক আছে, আর কি রিবোফ্লাভিন, these are three compounds commonly used as intercalating agents to visualize DNA. Now, after the job is done, the whole thing we throw in the sink of the laboratory, and it goes into soil, and then it gets mixed with the groundwater, and then it is pumped into RO water, and we drink that, or we cultivate. We use it for agriculture, and that same acridine orange comes to our body, right? And then when they make space between two bases, the DNA polymerase, while replication, they falsely read as 3 bases instead of 2. So they add a new base, causing insertional mutation. So that's why these intercalating agents are very harmful. And riboflavin in this aspect is harmless. The other two compounds are ঘুম থেকে ওঠ, তোকে বলছি দাঁড়া, যথেষ্ট বয়স হয়েছে, 5-10 মিনিট ধৈর্য রাখতে পারছিস না? কালকে ভোট, কিছু বললাম না, পকেট থেকে বোম টোম বার করতে পারিস, বস। কি বলছিলাম? Out of these 3 intercalating agents, riboflavin is harmless, but ethidium bromide and acridine orange are harmful compounds. That's why nowadays these 2 compounds are replaced by riboflavin or riboflavin, vitamin B2.